Hey everybody, today we're going to dig a little bit deeper into how DNA works, and we're going to take a look at specifically some very simple genetic combinations and kind of take a look at how genes work and how we get those from parents and how we determine which gene is expressed and figure out how we get the traits that we have. So all of this topic was the fascination of a monk from the 1800s named Gregor Mendel. He was an Augustinian monk and lived between 1822 and 1884. And his research, his work, he studied pea plants. He studied different traits within a pea plant, and he chose the pea plant on purpose because it is a type of plant that grows very quickly, and it's one that he is able to control the pollination from one plant to another so that he can make sure that he's getting the gamete from one parent plant and the gamete from another parent plant so that he can make sure that those are the two parents and he can see which traits that the offspring have, because that's what he was interested in. That's what he wanted to figure out is how is it that we have parents that have different traits, and then we get specific traits from both parents. And at the time that he was doing his study, the general idea was that our traits were kind of a mixture of our parents. So if you had a parent that has let's say blue eyes, and you had another parent that has green eyes, that if those two parents hadn't had a baby, then that baby's eyes would be a mixture of blue and green, which, according to Mendel's research, we know that that's not true. There are some traits that will have that type of behavior, and we'll talk about those at a later time. But today we just want to focus on the simple genetic inheritance traits that Mendel researched, and he figured out kind of how they worked. At his time, they didn't know that it was DNA that was doing all of the inheritance, passing on those genes. They thought it could have been protein, thought it could be carbohydrates. They weren't sure. All right. It wasn't until the early 1900s that they figured out truly that it's not protein that passes genes from one parent to the offspring. It's the DNA, and we'll talk about that at a later time. So Gregor Mendel studied these pea plants, and he studied specific traits within the pea plants. Those traits include things like the plant's height, include the flower position, pod shape, pod color, seed shape, seed color, and and the flower color. So through his studies, he figured out that parents will pass specific molecules onto the offspring that create these traits. And the offspring, the baby plant, if you will have one of the types of traits, one of the characteristics, or the other. For flower color, there were white flowers and there were purple flowers. And so when he would cross these plants, he figured out that the plants would either produce a baby that had purple flowers or a baby plant that had white flowers. So let's take a look a little bit deeper into how these simple genetic crosses work. Now, for each trait, we have what are called alleles. An allele is a version of a trait. So with Mendel's flowers, the pea plants, the flower color is either white or it's purple. And so those are the two alleles for that trait. You can either have an allele that codes for a white flower, or you have a version of the gene, an allele, that codes for a purple flower. among alleles, you will have what are called dominant alleles, and you'll have recessive alleles. The dominant allele is indicated by an uppercase letter. So with the flower color for the pea plants, purple is the dominant allele. So if a plant, one of those pea plants, had a dominant allele, then the flowers would be purple. If it had the recessive allele, the lowercase p, then those flowers would be white. And to make things easy, usually we'll use the first letter of the dominant trait as the letter we use to create what we call the genotype. I'll talk about what a genotype is here in a second. So within a diploid organism, remember last time we talked about meiosis and how through meiosis we form haploid gametes. Well, those haploid gametes, they have one chromosome for each trait, each gene in the nucleus. And so if it's a sperm cells are haploid. An egg cell is also haploid. So there you have two cells with one set of chromosomes each. So when we have fertilization, those two sets of chromosomes come together in the zygote, and now we have a diploid cell. So when we have a diploid organism, we will have two sets of chromosomes, one set from each parent. And so those chromosomes will contain different genes, and they'll contain different alleles for different traits. Those alleles can be either dominant or they can be recessive. So, as I mentioned before, we have what's called a genotype and we have what's called a phenotype. The genotype, if we shorten that down to just the beginning, just that prefix, we have the word gene. All right? The genotype is the set of alleles or the set of genes that organism has for a specific trait. For the genotype, there are three types of genotypes. One we call homozygous dominant. One is homozygous recessive. And the last one is heterozygous. As you can see up on the board, homozygous dominant means that both alleles for that trait are both dominant. For homozygous recessive, that means that both alleles are recessive. Heterozygous means that we have one dominant allele and one recessive allele. Let's look at those prefixes again. Homozygous dominant. Homo, again, we've talked about this earlier on in the year. Homo means that they're the same. So if we have homozygous, then both of the alleles are going to be the same. Homozygous dominant means they're the same and they're both dominant. Homozygous recessive means they're both the same, but they're going to be recessive. And then that prefix hetero means different. And so heterozygous means we have different alleles, one dominant, one recessive. Now, when we're writing a genotype, we want to make sure that if there is a dominant allele, we're writing that one first. So we'll write that capital letter first, and then the recessive allele, the lowercase letter, will be second. So that's the genotype, the set of alleles for a specific trait. Now, kind of related to that is what we call the phenotype. Phenotype, I like to shorten that down to that pH at the beginning, the physical appearance of a trait. So the phenotype is what we see is expressed through the genes. So we have two types of phenotypes. We have either a dominant phenotype or we have a recessive phenotype. If there is a dominant allele, then we will have a dominant phenotype. If we have a dominant allele, we will have a dominant phenotype. So if we have homozygous dominant, or if we have heterozygous as the genotype, then that dominant trait will be expressed. The dominant version of that trait, rather. If we have homozygous recessive, if there are no dominant alleles, then we'll have the recessive trait or the recessive version of the trait that will be expressed. So as I said before, the pea plants, purple flowers are dominant, white flowers are recessive. So if a pea plant has a homozygous dominant genotype, homozygous dominant would mean we have uppercase P, uppercase P. That means that we have a dominant phenotype. If that pea plant has a heterozygous genotype, that means an uppercase P and a lowercase p, then again, we have that dominant allele there, so it's going to be a dominant phenotype. The only way that a pea plant will have those white flowers is if the genotype is homozygous recessive. We do not have a dominant allele there, and so we'll have the recessive phenotype. And that's white flowers. Now, if we know the traits of both parents, then we can start to make predictions about if the two parents had an offspring, what's the probability that those offspring will have certain traits. You've already covered these in, for most of you, 6th grade. And these are called Punnett squares. And so you may remember these. Punnett squares help us to predict the probability of producing offspring that have certain traits, have certain genotypes, when we're looking at the genotypes of two parents. So here for this example, we're going to have both parents will be heterozygous. No, I'm sorry. Both parents, one parent will be homozygous dominant, one parent here will be homozygous recessive. Now, we'll write one parent across the top, we'll write their genotype. So we're going to put the homozygous dominant genotype across the top. And then the other parent, we write their genotype down the side. So we're going to put the homozygous recessive genotype down the side. Now what we do is we take those and we will write the first allele of the top parent. We're going to write that in both squares in that column. Second allele in the genotype of the top parent, we're going to write that allele in both squares in that column. Then we're going to do that same thing, dragging the alleles across for the other parent into each row. And so once we've completed our Punnett square, we see if we have a homozygous dominant parent crossed with a homozygous recessive parent, every single one of the offspring will be heterozygous. And so from here, we know that every single one of the offspring will have a dominant phenotype. And that's what we use Punnett squares for, is to predict the outcome of genetic crosses. This here is what we call a monohybrid cross. Mono, that prefix means one. So we're looking for a cross between the genes of parents for just one trait. At a later time, we'll talk about and we'll see how to look at the probability of offspring, the traits in offspring, when we have two traits that we're looking at. So here I have a very simple monohybrid cross. What we can do with this is we're going to figure out, and we're going to calculate the percentage of probability of producing each of the three different genotypes. So we want to know, if we have two heterozygous parents, what's the probability that the offspring will be homozygous dominant? What's the probability that the offspring will be homozygous recessive? And what's the probability that the offspring will be heterozygous? So when we complete this cross, we have two heterozygous parents. What we find is that 25%, or 1 out of the 4, are going to be homozygous dominant. That's 25%. What we find is that 50%, or 2 out of 4, will be heterozygous. And then finally, 25%, or 1 out of 4, will be homozygous recessive. And we can see that here. 1 out of the 4 is homozygous dominant. 2 out of the 4 are heterozygous. And then our last one is homozygous recessive. So if these two parents have 4 offspring, probability says that 1 of them will be homozygous dominant, 2 will be heterozygous, and 1 will be homozygous recessive. Now we can do this for any trait that follows what we call Mendelian genetics. Like I said, this is very simple genetic crosses that follow that dominant versus recessive behavior. There are special circumstances that we'll talk about in another video. But for now, these are the crosses that I want you to be able to complete. And you should be able to calculate the probability for specific traits in a monohybrid cross. Now I've posted a follow-up task in Google Classroom. Go there, complete that task, make sure that you submit, and we'll see you next time.